Hello friends, welcome back. In this video, I'll be solving gate-xl MCQs and MSQs of biochemistry paper 2024. In the previous video, I have solved few questions from this biochemistry paper 2024. In this video, I'll be solving 4 questions each carrying 2 marks. And in the upcoming video, I'll be solving the remaining questions from the biochemistry paper 2024. Let's solve. Question 36. In this question, they have given two columns. Column 1 contains the metabolic intermediates and column 2 contains the corresponding amino acids. We have to match which metabolic intermediate forms which amino acid. So let's match. Alpha ketoglutarate forms glutamate amino acid, ribose biphosphate forms histidine, 3-phosphoglycerate forms serine, and phosphoenol pyruvate forms phenylalanine. So according to our match, the correct answer would be option A, that is P2, alpha ketoglutarate forms glutamate. Q1, that is ribose biphosphate forms histidine. R5, that is 3-phosphoglycerate forms serine. S4, that is phosphoenol pyruvate forms phenylalanine. Let's move to the next question. I'll mark the answer, that is option A. Let's move to the next question. Question number 37. In column 1, they have given molecular properties of biomolecules. In column 2, they have given the biochemical separation methods. We have to match molecular properties corresponding to biochemical separation methods. Now, these are the properties of biomolecules. For example, let's consider protein. We have a mixture of proteins. So, based on solubility, we can separate mixture of proteins by salting out method. Based on ionic charge, we can separate by isoelectric focusing. Based on polarity, we can separate by reverse phase chromatography. Based on molecular size of proteins, we can separate by ultracentrifugation. So like this, we can separate based on properties. So according to our match, the correct option would be C, that is P3, solubility by salting out method, ionic charge, Q4, that is ionic charge by isoelectric focusing, R1, that is polarity by reverse phase chromatography, S2, that is molecular size by ultracentrifugation. So correct option would be C. Let's move to the next question. Question 38. Which one or more of the following statements are correct regarding the electromotive force generated by electron transfer chain? Options. It is used for synthesis of ATP. It is not used for active transport process. It includes a pH gradient component. It does not include an electrical potential gradient component. This question is of MSQ, multiple selection question. Two options are correct and two options are incorrect. ETC, electron transport chain, which occurs in mitochondria. In mitochondria, in inner membrane, in inner mitochondrial membrane, there are a series of protein complexes which are embedded, complex 1, 2, 3, 4. In ETC, there is a flow of electrons from one complex to another complex. During this flow of electrons, there is a flow of protons from matrix to inner membrane space, which creates a proton gradient across the membrane. Proton gradient is a pH gradient, and due to this proton gradient, there is a there creates a difference in proton concentration across the membrane and the charge across the membrane, which leads to electrical potential gradient across the membrane. There is an enzyme, that is ATP synthase, which is also embedded in the inner membrane inner mitochondrial membrane that controls the proton gradient to synthesize ATP. This proton gradient, which drives ATP synthase, that uses the energy of proton flow to synthesize ATP from ADP. So, option A, it is used for the synthesis of ATP, and option C, it includes a pH gradient component. Are both options are correct. And B option, that is, it is not used for active transport process, which is absolutely wrong because it is used for active transport process. And option D, it does not include an electrical potential gradient. As I told, electrical potential gradient is generated when there is a proton concentration and charge differences across the membrane. So option A and option C are correct. Let's move to the next question. Question 39. Which one or more of the following statements is are correct regarding the transport and retention of proteins in different cell organelles? Options. Mannose 6 phosphate residues are involved in targeting proteins to lysosomes. Transport of proteins into the mitochondrial compartment is aided by positively charged amino acid residues at N terminus and internal hydrophobic segments. The retention of protein in the ER lumen requires the Kedel sequence motif at the C terminus. Nuclear proteins are transported in an unfolded conformation, and the nuclear localization signal sequence is subsequently cleaved by peptidases in the nucleoplasm. In this question, three options are correct and only one option is incorrect. Option D is the incorrect option. Nuclear proteins are transported in a fully folded conformation. It will not transport in an unfolded conformation. The nuclear localization signal sequence is never cleaved by any of the enzymes in the nucleoplasm. Option A, B, C are correct. Mannose 6 phosphate residues are cleaved in targeting proteins to lysosomes. Transport of proteins into the mitochondrial compartment is aided by positively charged amino acid residues at N terminus and internal hydrophobic segments. These positively charged amino acid residues and the internal hydrophobic segments are the two mitochondrial targeting sequences. This amino acid residues at N terminus is cleavable, but internal hydrophobic segment sequence is uncleavable. The retention of protein in ER lumen requires Kedel sequence. Kedel is lysine, aspartate, glutamate, and leucine. This sequence is important for the retention of protein. If this sequence is cleaved from the protein is ready to export from the ER lumen. So for the retention of protein, Kedel sequence is an important motif to be present in the protein. So option A, B, C are correct and option D is incorrect. Thank you. This is the end of the video. We'll be seeing in the next video.